Okay, welcome to Statistics 115, 215 again, and this is the first lecture after spring break, and we are doing full teaching on Zoom. I cannot see any of the students in class, so this will be quite an experience. I have by default muted every participant, but if you have questions, you could unmute yourself and ask questions, okay? So today we're going to talk about long-range chromatin interactions and regulatory network. Earlier in this module, we talked about DNA methylation, nucleosome positioning, histone modification, and chromatin accessibility. So today we're going to talk about higher order chromatin interactions. So in recent international projects on looking at how genes are regulated, there is a major consortium called ENCODE. They generated a lot of ChIP-seq data for transcription factors, histone modifications. There are also a lot of chromatin accessibility data in many, many cells and tissues. And they look at how many percentage of the genome are really potentially functional or contain regulatory sequences. As you know, in the human genome, coding sequence only accounts for about 2%. But the remaining genome, based on the ENCODE publication in 2015, about 13% of the human genome are likely to be devoted to enhancer function in at least one of the cell types that they have profiled. So you can imagine there are many, many binding sites residing in the genome, and they might be functioning in different cells to regulate different genes. So the question is, how do enhancers really target specific genes from a distance? And there has been increasing evidence to suggest that very often the enhancers can be looped to the promoters of the genes, and the transcription factor that's bound in the enhancer location can help recruit polymerase to the promoter or help push polymerase to kick off the transcription elongation process. And so this kind of got people interested in how chromatin is looping with each other to interact in order to start gene expression. And so if we look at chromatin in a cell, so we know that the DNA or the chromatin is packaged into the nucleus, and the DNA is actually very, very long, and there are interactions here that that happens in the local structure here. And you can imagine filling the human genome into a nucleus is similar to filling a string that's about 44 miles long into a basketball. How do you package it well so that the correct interactions can happen in order for the correct enhancers to activate the correct gene? This is really quite challenging, right? And the cell over evolution has really figured out how to do this well. Initially, in evolution, there are simple genomes. They're very small. For bacteria, coding sequence is 80%, right? And then you have more and more complicated genomes like yeast, like C. elegans, and then there are humans, right? Or vertebrates or mammalian genomes. And the genomes gets bigger and bigger, and in order to package it well and still activate the genes at the right time, the chromatin needs to package correctly. And so this is very, very important. And in order to really understand the promoter and enhancer and how they loop, how they interact to regulate gene expression becomes a very interesting question. And there has been very well-known example where promoter enhancer interactions are important, and when this is disrupted, it can really cause important diseases. So there are published papers, for example, in the left side, it's known that, for example, there can be some congenital malformations. For example, if the enhancer, so on this left side, if you can see the arrow, there could be enhancers that's green and a promoter that's yellow. And in normal conditions, these two don't interact with each other. Therefore, this yellow gene is turned off. But in some mutations, you can see that for some reason, the enhancer is now looping to the promoter of the gene, and this gene become activated. And then that could create important bad diseases. In this case, people can either have fewer fingers or too many fingers because of whether the gene is turned on or off. And on the right side, this is related to cancer. For example, MYC is a very, very important gene. Many cells express MYC, but it has to be expressed at the right level. In many cancers, MYC are expressed at much higher level, and it's kind of a amplifier for overexpressing many important genes in the cell. And MYC is actually in a very gene desert region. There are not many genes near MYC. And there are many interesting enhancers in here. And depending on the different cell conditions, sometimes it's being activated by an enhancer nearby. Sometimes it's regulated by an enhancer that's even further away. And sometimes so you can see in leukemia, it's being activated by a cluster of enhancers that are really far away. Whereas in colon cancer, it's being activated by another cluster of enhancers, but that appear in the opposite direction of this gene. And so depending on which enhancer is looping with MYC to activate the gene, it actually can cause different diseases as well. And so in order to really look at the chromatin interactions, there are techniques developed. The early technique is called chromatin conformation capture, or it's called 3C because it's chromatin conformation capture, right? So you can imagine if there are chromatin interactions. In this case, the blue is one region of the chromatin, and the red is another region of the chromatin. They probably reside far away in real genomic locations, but because of interaction, looping, they might be interacting with each other. In the 3C experiment, we first use cross-linking to cross-link them together, and then use restriction digestion. By the way, the arrow means that the chromatin is pretty long. It doesn't just stop here. They continue to link to the rest of the genome. And so we can then use restriction enzyme digestion to cut this into smaller, they just kind of cut based on recognition of things that probably on average happen every few kb. And so after the cut, you can see this is being cut to a smaller region. And then we can use a enzyme to ligate the fragments that are close by to each other. So you can see here, the red and blue, because of their physical interaction or physical proximity, are being linked together, even though in genomic distance, they used to be, they are pretty far away, but now they are being linked together. And then once we do the cross-linking, reverse cross-linking, these two are now right next to each other. And then if we can use a specific approach, so in this case, the linkage location are also tagged with a biotin. So we just enrich for the ligation points in the genome. So we eliminate all the genome that's not ligated. We only enrich for the ligated fragments, and then use high throughput sequencing to sequence them out. What you will see, so the 3C is in this case, well, in the early days, we just use a PCR primer. You can see the little blue arrow and the little red arrow. It means that like if there's no looping, this two PCR primers will amplify a region that's much further away. But if you do the HiC experiment and these two regions, if we do the 3C experiment and these two regions are interacting with each other, after the ligation, using PCR, we are able to amplify a region that's much, much shorter. That means that these two are interacting. So the original chromatin conformation capture, we have to know what is the left region, what is the right region, and we specifically ask, is this A region interacting with this B region? So it's a one-to-one interaction. You have to check on one pair of regions at a time. But it's still already a technology improvement that you can actually see whether chromatin, far away chromatin regions are interacting with each other. And then this is being improved to 4C, which is you have one anchor region one region you want to see, does it interact with this region or next region, another region? So it's a one-to-many. So you can look at one particular region, how it interacts with all the nearby regions. So it's one anchor against all the nearby regions. And people start to use high throughput sequencing or using early days array approaches. That's called 4C, which is one region to many. And then there's 5C, which is if you care about the whole 1 megabyte region, you can look at all the pairwise interaction within this megabyte. So this is kind of many to many interactions. But what we're going to talk to you about today is called HiC, which really captured the interaction fragments in the whole genome. And so it works like this. Again, we use cross-linking first. So if there are chromatin loops or chromatin interactions, they are being cross-linked together. And then we use restriction enzyme to cut the chromatin to smaller fragments. And then we fill the ends with the biotin and then use enzyme to ligate the things together. And so you can see here, anytime there is an ligation, they are being labeled with this purple biotin. Therefore, you can, after the cross-linking, you can see in this case, after the cross-linking, we can pull down only those DNA fragments that are formed through the ligation. And then we could adapt high throughput sequencing adapters to sequence them out specifically. And then based on the left read and right read, where they map to the genome, we will know, oh, this fragment is interacting with another fragment in the genome. And so this is really counting the interactions in the genome, theoretically all against all. Of course, it depending on how deep you sequence. The very first, earlier HiC resolution wasn't so good. They could only map reads that are interacting with, like whether this 40 kb of region is interacting with another 40 kb of region. So the resolution is not so good. In order to really have the resolution to, for example, 20 kb, we have to sequence 4 times. And then if you want to make it 10 kb resolution, you have to sequence 16 times. So you can see if you want to get better resolution, you have to exponentially sequence the HiC experiment. So it does get quite expensive in here. But with this type of experiment, we can then look at the chromatin interactions. For example, this is the first HiC in 2009, where they look at the contact map. So you can see this is kind of on the X and Y axis is also again from chromosome 1 to chromosome X, and on the y-axis is also chromosome 1 to chromosome X. It's kind of symmetric. We're asking how much do we see things interacting with each other. And so there are two things.
I have a question.
Yes.
Can you go back to the previous slide?
Mm-hmm.
So when you you use the biotin to pull down and then you sequence these fragments at the end. So like you're sequencing basically the parts that got cut with the restriction enzyme, but how do you actually tell where the actual interaction points were? Because
sometimes they don't know exactly where this junction is, because high throughput sequencing only sequence the left and right.
Right. Yeah.
And that's so basically that's also determining your HiC resolution, because restriction enzyme always recognize like a 4 nucleotide or 5 nucleotide to make a cut. And at the end, when you kind of do high throughput sequencing, it's also kind of how much you can see. So basically, they don't necessarily know, well, I guess you can at least know there is a sequence, right? Because the restriction enzyme know where to cut.
Yeah.
But at the end is you sequence the left of this fragment and the right of that fragment. You only know roughly this left chunk is interacting with that right chunk.
Oh, okay. But you don't know where the actual like interaction is, like where where the DNA was actually touching.
That's right.
Okay.
Yeah, so the resolution is limited by your sequencing depth. It also is limited by the specific restriction enzyme you have. That's why even the best contact map only have 5 to 10 kb resolution.
Okay.
Early days is like 40 kb resolution. Okay, so roughly you can see each chromosome is this little square. And within this, you can already see, so by the way, if it's really red, it means there are more interaction. But if it's, if it's white, it means actually it's unmappable, because the end of the chromosome, usually there are many repeats you can't map. But if it's kind of orange, it means there's not very, well, light orange, it means there are not so many interaction. But if it's kind of darker or red or orange, it means there are more interactions. And so at the very first glance, what you can probably see is that there are more interactions within the chromosome. And also there are more interactions when things are close by. That's why the diagonal is super red. It means that when things are close by, they do have more likely, they're more likely to interact. Whereas when they are further apart, they are not likely to interact as much. But you can see there are some slight differences how things can divide up, right? How they interact. But that's kind of the first level of data we get. And in order to really get a good interaction map, we need to make sure that the data is normalized correctly. And in order to do this, we first need to make some assumptions. By the way, HiC experiment, so far, you cannot do this, well, at least it's very, very difficult to do this on primary tissues. Most people do HiC in, say, in cell lines where you have like 100 million cells. And so the HiC contact map is kind of the average behavior of interaction in many, many different cells. And so supposedly within the HiC, so we are doing, in this contact map, we're looking at the pairwise interaction, right? So you can imagine, supposedly there is one region that's A, it can interact with 1, 2, 3, 4, 5 different regions, whereas another region that's B, it's only interacting with location C. What you will see is that, for example, maybe in one cell, A is interacting with C, so 3. In another cell, A is interacting with 1, and another cell, A is interacting with 2. Yeah, so you can see here, in different cells, A might be interacting with a different partner. Whereas in this B location, it's always interacting with with 6, right? And so at the end, what you get is these A-B locations, how they interact with each other. And when you sum them up with many, many cells, what you will see is that this A location, you can see some streaks in here. This A is interacting with many, many places, whereas B is really specifically interacting with this 6 location, right? So it depends on how you really capture this. The specific interaction, if it's really like this, it's actually okay. Unfortunately, very often, what you see is, these type of streaks are not necessarily due to real interactions, but probably due to restriction, enzyme cutting, or sequenceability. Some regions are easier to sequence than others, and other potential biases. And therefore, we need to really make sure that when we analyze this HiC contact map, it's normalized correctly, so that if some regions are more easier to sequence, you don't give it too much emphasis, right? So we want to be fair. And so this requires this big matrix of contact map to be normalized. And so, for example, you can see here, basically we want the columns and rows to roughly sum up to equal levels overall. So, in this case, if we start with an initial contact map of just read mapping to this location, you can already see that there are very different level of total either column or row sums, right? So in this case, for example, first we can calculate a row mean. And you can see here the first row has a higher mean than the second row. And so in order to really normalize the interactions, what you can see is, what you can do is to divide so in all the numbers in the first row by the mean of this row, right? So, and the second row, all the numbers you can divide by the mean of the second row. So just to make sure that the interactions you see are reasonable. And so by the way, so the first number 8 is actually being divided by the mean of the first row and the column of the, sorry, the mean of the first column. Does that make sense? And so this way, you want to make sure at the end, the columns and rows are more or less comparable. That's how you want to kind of normalize it. And you kind of iterate this until at the end, the row sums and column sums are more or less comparable. I do see some questions in here. Let me see whether I can look at the chat window. Is the interactions between chromatins mediated by transcription factors? That's a very good question. Theoretically, yes, but depending on the HiC resolution, currently people are not very good at really looking at specific TF enhancer promoter interaction loops, either because they haven't sequenced deep enough or depending on the specific transcription factors, these type of loops may not be very stable. Therefore, HiC has not really been very successful at capturing promoter enhancer loops yet. Another question is, wouldn't we want the row sum to be normalized by the sequencing depths? Here we are talking about all the interaction in a single experiment. We're not even comparing different HiC experiments yet. This is a HiC contact map for different regions in the same for the same experiment. And so we just want to make sure if there are sequencing biases, you want to kind of normalize it out. Okay.
I had a follow-up question to the first question. Then what does it mean for HiC to claim there is an interaction?
So
does it mean the sequences interact with each other, or there's a TF factor that acts as a bridge between the two locations?
In the early days, when people were doing 3C, they were hoping to look at the specific promoter enhancer interactions. But at that time, you don't really need to get a quantitative result. You just need to see in a particular cell, something that was supposed to be far away. Do you even see a short band? That means that they are kind of interacting. It's kind of black and white interactions. But then people want to know, oh, this is great, but you don't want to just do A and B in one cell. You know, you want to do all the potential interactions. And so people are trying to increase the throughput. And as you increase the throughput, they also want to get a more quantitative interactions. And what at the end, what they captured is what they were able to see is just this chunk of chromatin is interacting with this chunk of chromatin. And you can see when you're talking about a 40 kb resolution, you just know roughly this big chunk is closer to that big chunk. Very often the chromatin interaction happens really within the 40 kb, and you just don't have enough resolution or enough quantitative data to really give you a confident interaction to say that 20 nucleotide is interacting with the promoter of a gene yet.
I have a follow-up question as well.
Yeah.
So when you're doing the cross-link between the DNA, how would you, like, what's the specificity of it? And also if these two chromatins is already been linked together by the transcription factor, like the CTCF, how would the cross-link, I suppose it's a protein, also target and cross-link those two?
Yeah, so remember later on, we also do decross-linking. So if you don't do a ligation, using the restriction enzyme to ligate this together, after decross-linking, you would lose that interaction.
Okay, but like how, if there's already some transcription factors that's linking these two chromatins together, would the cross-link protein that we added able to link those two like target that site again, or?
Yeah, so for example, you can see here, supposedly this blue region is bound by one transcription factor, and this orange region is bound by another transcription factor. Because of the two transcription factors might be interacting, these two pieces of chromatin might be close to each other, right? And if you use cross-linking, you just try to stabilize the interaction a little better so that it can withstand the cutting and the ligation, right? To just make sure that they stay together as you do the reaction. But eventually, you do decross-linking, and then you would lose that piece of protein-DNA interaction, and also protein-protein interaction, and the DNA-DNA interaction. And so the ligation will make sure that whatever was captured before, you will still be able to sequence it out at the end.
Okay, thank you.
Yeah. Yeah, so this type of experiment is very powerful, and so many scientists in the world started doing these chromatin interaction maps now, or these type of experiments. And there are also international consortium called 4D nucleome program. So 3D means you want to see how, so you can imagine 3D is kind of chromatin interaction, but the fourth dimension is the time. You want to see what happens in development, what happens in disease, what happens in cell cycle, and things like that. So these are the international consortiums that are generating these type of HiC maps. And so, and then that's the rough experiment. We will show you what are the major findings in the next part. Are there more questions?